Hi, good morning, everyone. We'll wait for a couple of minutes more. Hi, good morning, everyone. I'm Dr. Devlina Bandyopadhyay. I am a scientist at Merck Innovation Lab. I welcome you all to Merck's monthly webinar series. The topic for today's webinar is Multiplexing Immunoassays, Unlocking High-Throughput Precision in Biomarker Detection. Now, today's session would be for a duration of an hour with 10 minutes question-answer session at the end. During the course of the webinar, we would also be posting poll questions in the chat box. So I would request the audience to kindly participate. The fastest person to correctly answer wins exclusive Merck merchandise. So we have two speakers for today's session. Our first speaker is Dr. Deepak Deep. Dr. Deepak finished his PhD from ICMR National Institute of Pathology. After a brief postdoctoral experience investing host immune response following infection with drug-resistant Leishmania parasite at ICMR, Dr. Deepak joined Merck Innovation Lab as a scientist. In his current role, apart from conducting hands-on and digital training courses for life science researchers, he also provides extensive application support for multiplexing services, specifically on the Luminix assay platform. So over to you, Dr. Deepak. You can please proceed with your session.
Thanks, Devlina. And please confirm if slide is visible and audible.
Yes, Deepak, your
So,
slides are visible as well. You're audible as well. Please proceed.
very good morning. Welcome you all. And today, the topic of discussion in the webinar is the multiplexing immunoassays, unlocking high throughput precision in biomarker detections. So, before we start on this topic, we'll discuss actually what is biomarker actually is. So biomarker, we all know that any molecule that can be quantifiable and measurable in our biological samples and which indicate any normal or the abnormal processes happening, biological processes happening inside our body. It may be usually the proteins, any cytokines, chemokines, interleukins, or any and the nucleic acid molecules. So introducing to the what is multiplexing immunoassays. So multiplexing immunoassays actually tells us about the detection and the quantification of multiple analytes, like it may be your proteins, cytokines, or any antibodies in a given biological samples, which facilitates efficient analysis of multiple targets in a single sample in a single reactions. It is an high throughput and high sensitive analyte detection techniques which facilitates researchers to investigate the multiple processes happening in a cascade in any biological entities. This is the workflow in the multiplexing. So we should have a samples, then we mix it with the and multiplex beads and distribute in the array, and there are multiple, we can investigate multiple targets for which we have a individual capture antibodies coated on the beads and the secondary detection antibodies we pull together, and we can proceed with the simultaneous detection by running the assay, and we can finally process the data for analysis simultaneously. So why there is a need of multiplexing in the biological research is it saves time, it saves the sample, as well as the cost to the researchers. Suppose we have 10 different proteins to detect in the given clinical samples, what we are having in the ongoing projects, and we have 40 different samples. So if we go for by performing the conventional ELISA technique, so we have to perform 10 different assays, will consume 10 different plates, and it will take lot of times, approximate 60 hours to finish this project. But when we proceed with the multiplexing technologies, what we can do, we can achieve the similar target in just one assay, which not only will save our plastic consumables and it gives us a sustainable approach, but it also significantly saves our time, cost, labor, as well as the it is on high since it is on high throughput, so it gives us the precise answers of what we are investigating in our project. Why the sensitivity matters in this biological complex samples is, so we have altogether 30,000 to 40,000 genes, and we have multiple thousands of proteins present. Most of the proteins are present in very low abundance, which goes undetected when we proceed with the conventional immunoassay to detect as a biomarker. In a high throughput assay, it provides us the large dynamic range, high sensitivity, and precision so that we can screen out the several thousands of possible and target molecules which we can screen in our biological samples which were previously undetected, so that we can focus on our molecule of interest. Further, we narrow down their detection, and we can find the significant number of molecules which may play an important role in detecting our pathophysiological symptoms present in the happening at a real time. Okay, and at the same times we can quantify the samples. We can quantify the multiple samples, multiple analytes in the large throughput of samples in a single reactions. That is why the sensitivity during the detection of this biomarker or the protein becomes crucial whenever we are running several high demand in any project or investigations in large cohort of sample, particularly for the clinical samples or the any translational research. So their applications are very broad. Whenever their applications are very broad, and in the multiplexing, we can focus at the same times in the inflammation immunology, neuroscience, and oncology, which by performing this multiplexing, not only we can detect the low abundance biomarker crucial for understanding the disease mechanism or the development of pathophysiology and the any tumor progression studies, we can more focus on our the target molecule at the same time. And it is essential we can further narrow down the potential biomarkers for early stage detection, early stage development of any neurological disorder or the cancer tumor progressions. Most of the physiological conditions or the pathologic conditions, many of the biomarkers or many of the molecules undergoes down regulations. If those downregulated molecule we may escape while we perform the immunoassay by conventional method, but since once we approach the high throughput and multiplexing assay platform, we can even measure the low abundance molecule present in the given sample in more precise with accuracy, with more accuracies. Okay. Sometimes when we do the conventional ELISA or conventional immunoassay detection techniques, we have several challenges. Okay, there are so many of interfering molecules, so that we can have a false positive or false negative result. To rule out this, what we use in the multiplexing technology platform is we use some surrogate matrices. What are those surrogate matrices? These are the simple ionic buffers which have a similar ionic strength and the pH conditions similar to the biological fluids, but it lacks the complex proteins and the complex component which is specifically present in your samples which you are going to analyze or investigate. The limited sample volumes in the conventional way of doing ELISA or an immunoassay, we need a large volume of samples. But when we perform the multiplexing samples, we need very small amount, almost 20 to 25 microliters of any samples. It may be any body fluid. In pharmacokinetics and the pharmacodynamics study, whenever we are investigating the after effect or the treatment process like ADME, absorption, distribution, metabolism and excretion of any drug molecule. So how the treatment response is going on. We can measure the several orchestrated molecules playing important role in cascade by performing the multiplexing assays. So we have now several high throughput platform where we perform the multiplexing assay is called as an XMAP technology. So what is XMAP technology here is X is your unknown, M stands for multiple, A stands for analyte, P for the platform. So we have a multiple analyte platform where we can go ahead with the detecting multiple biomarker molecule to screen or to investigate the conditions, disease conditions, or any pathophysiological symptoms during the investigations. So what XMAP technology actually is, it is a bead-based technology where we use the polystyrene beads. On an average diameter of these beads are the 6.5 micron. And these have a conjugated coupling molecules. Most usually it is a carboxylic acid, carboxylic group that plays an important ligand molecule through which our analyte molecule joins. Okay, this molecules provides a very sufficient surface area to conjugate almost 10 to the power 7 to 10 to the power 8 different biomolecules on a single bead, right? So what actually we do in the bead map technology is we have several analytes, right? For all the analytes, we have a dedicated microspheres bead. how we can differentiate one analyte from the other is the bead which these microsphere beads are differentially coated with the red dye and the infrared dye. So we have a various combination of this red dye and infrared dye, which makes this bead differentially distinct from one analyte to the other analytes. And altogether, we can have several 50 to 100 to maximum of 500 differential color coding concentration on the bead. Based on those color differential color gradient color of this red and infrared dye, it provides us a spectral address to the bead, which makes it very specific to the analyte. Okay, this on this bead, usually this bead is coated with our capture antibodies, which is very specific to the analyte. In the XMAP technology, this magnetic bead is often coated with the capture antibody, and this bead can be interrogated by the red laser, which excites as the 638 nanometer of wavelength. And through the primary antibody, and we have another antibody, we call them as the detection antibody, which is fluorescently conjugated with some fluorescent molecule, and it is streptavidin phycoerythrin biotinylated detection antibody. Our analyte, that is a protein of interest, or is sandwiched between the primary antibody coated on the beads and the secondary antibody. So they form an immune complex. Okay, this immune complex we are going to detect by on this multiplexing platform. So how we detect this immune complexes is by using the Multiplex technology. It is a combination of fluidics and the optics approach. In the optics approach instruments, we have two different types of laser, the red laser and the green laser. So whenever the bead is passing through this instrument, through this laser, the first interrogation of bead happens with the red laser, which actually interrogates the bead and provides this and investigate whether the bead is in the specific region or not. Once the red laser interrogates the bead, further it encounters the green laser, which actually measures the fluorescence intensity released during the passing of the bead and quantify the analyte present or bounded on the bead. So there are several beads will be passing in the narrow stream, and each bead will pass one after the other and interrogated by both these lasers. Okay, the green laser detects the amount of target bounded on the uh, microsphere beads. So this is a very sophisticated and sensitive technique, which which allows researchers to detect multiple target proteins bounded on the beads based on the bead region assigned for the particular analyte. And for every particular analyte, there will be a specific capture antibody and the detection antibody cocktail present in the reaction mix. In the XMAP technology, we have several series of instruments through which we perform this assay. It's like the LX200 system, Luminex 200 system. With these instruments, we can go investigating maximum of 100 different analytes, and it takes usually 40 to 45 minutes to complete the assay. We have a FlexMap 3D system where we can increase our analyte range up to 500 different plexing level. And this is very fast. It follows very fast kinetics, and the reaction and the complete assay usually takes 20 minutes. We have a XMAP IntelliFlex systems, and XMAP IntelliFlex system, we have a two variant. The first one is that is the research use only are XMAP IntelliFlex RUO. And another is the DRSE model, where we have one additional laser which facilitates us to investigate the several subtypes of any analyte present. The maximum the capacity of this instrument is we can go of maximum of 500 different analyte plexing level, and it takes 20 minutes to perform the assay. One more advantage of this system is we can perform the 96 well assay plate as well as the 384 well assay plate detection system. So we can increase our assay range. We can increase our sample quantity from 96 to 384. These are the very high throughput and the sensitive platform, because since it is a laser based and it follows the fluidics followed by the optics detection method. Now we can have a poll questions, Devlina.
Yes, Deepak. We have posted the poll question in the chat box.
Okay.
To the audience, we request you to please participate in the poll question. So let me read out the question again here. What is the main advantage of multiplexing immunoassays compared to single-plex assays? Option 1, they require more sample volume. 2, they allow simultaneous detection of multiple analytes in a single run. C, they are less sensitive. D, they only detect one analyte at a time. We have 88% of answers coming from they allow simultaneous detection of multiple analytes in a single run. That is option B.
Excellent. That is the correct answers.
Okay.
So I hope that audience are enjoying the sessions. We got a very good response from the audience in the poll questions. Now we'll move to the next section, that is how we perform the analysis once we have finished the multiplexing assay, right? For any immunoassay, the analysis of data becomes important because we never have a linear response of the measurement of the biological responses. Okay, so because since we know that the biological system is a very complex system and several molecules have an interplay, and this is how it becomes more complicated. A signaling system or the model to be used in an immunoassay. In the multiplexing assay, we have to interrogate the several linked or the multiple interrelated pathway. So it becomes very crucial how we are investigating the data. So we have usually in the any immunoassay data, we never get a linear curve. Or most of the time we get the sigmoidal nature of the curve, which makes it more sensitive and which provides us the large dynamic ranges so that we can very correctly measure and quantify the range of the analyte present in the sample. Okay. In the sigmoidal curve, we often test for the goodness of the curve fit. What is curve fit? Curve fit is parameters where we investigate the standard curve and the quality control steps in the reactions, and we interpolate the unknown sample data, how an unknown concentration of the analyte is fitting well into the curve. So we uh, while we perform the assay, we run the different dilution factor of the standards as well as the quality control steps. And we investigate the recovery and the spike of the analyte present in the assay sample. So what is the recovery? Recovery is the ratio of the observed concentration and the expected concentration of the analyte present, and it is multiply with, if we multiply with 100, it gives us the percent recovery. What are the spike in, why spike and recovery is important is it allow us to investigate the accurate measurement of the concentration of the analyte present in the biological samples, and we can negotiate about if there are any cross reactivity happening in the during the multiplexing. So we minimize the background and the noise signal by spiking and through obtaining the recovery of the standards, and it measure the concentration compared to the expected concentration or the spiked concentration of analyte present in the assay. Now we can process the raw data simple by on the Excel sheet, but since nowadays we have several online tools or several dedicated software which gives us a very handy data and error-free final analyzed data. One of the known software for analyzing the multiplexing data is the BELISA, right? So what BELISA actually means is BELISA means it's illuminate in the Swedish. Okay, what we may do in the BELISA analysis is BELISA is a curve fitting software that allow optimization. We can visualize the data, and at the same times we can process our data without any error. We can fine tune our data, that is called the data hygiene or the experimental hygiene, so that we can neutralize, we can minimize the background signal, noise signal, and it gives us the high throughput increased signal by noise ratio. These are very user friendly software which gives us the very quick analysis of our sample in a very large cohort of the sample or the high throughput analysis. So once we open the software, so this is how the blank page appears. We can go through the file, or we can directly drag the file to for the analysis into this location. And once once we run this software, this is how the standard curve appears on the screen. So it is a perfect sigmoidal curve, and the vertical dotted line indicates us about the LLOQ value. What is LLOQ here is lower limit of quantifications. So we have a large dynamic range 3.5 fold to 5.5 fold. So we can better investigate the concentration of analyte in the given sample or in an unknown sample, and we can better compare with the standard curves generated through this software, right? So this software is compatible with any of the Luminex instrument, LX200, FlexMap 3D, IntelliFlex, MacPix, along with the SMCX Pro Reader also. And we can use the data generated by several other platforms like Bioplex Manager, multi-analyte export file. On the entry screen, we can see there will be several sections. In the left sections, we can check for the plate, what we are going to investigate. On the below, we can interpolate the experimental data. So we can drag the standard, control, and the unknown samples, and we can run the software. On the right panel, we can have a for the data optimization, we have a curve fit software. So we can we can select the curve optimization, and we can double click on the curve optimization. Quickly, it will optimize the standard curve and gives us the value of the different analyte and the standard value present in the and during the analysis. So this curve fitting software analyze data very quickly, and it is based on the 4PL or the 5PL logistic parameters, which very efficiently and quickly fine tunes our data. By using this software, we can also do some manual adjustment in the curve fit by double clicking on the fit optimization curve, and selectively we can also see where are the flags value in the standard or control or in any unknown sample. So the flag data will be indicated in the triangle, a flag triangle like yellow color or in the red color, which tells us about the value which is going beyond the dynamic range. It may be either below the lowest limit of quantification, that is called limit of detection, LOD,
Hi, Deepak.
or
Hi, Deepak. I'm sorry to
yes,
interrupt
yes,
in between.
yeah,
So, in the interest of time, we need to move on to the next
yes, yes, yes.
segment.
So I
So,
think
can we please wrap up in
yes,
next couple of
yes, yes,
minutes?
yes, yes, yes,
Thank
yes,
you.
yes, sure. So it will take only 1.5 minutes more.
Sure, sure.
So we can investigate the value of the unknown below the limit of detection also and the upper limit of quantification also. So we can quickly investigate what are the percent CV and the percent recovery we are getting through the standard curve, right? So this is a very efficient and handy tool, this Beliza, which provides us the curve comparison tool, so we can compare the data at two different time points, like inter-assay data and the intra-assay data. We can also do the parallelism study. What is parallelism studies? So in the two different set of experiment, we can compare the standard curves, how accurately these standard curves are matching from experiment 1 to experiment 2. Also, we can investigate the relative potency test between the experiment and the parallelism test of the any has any biological definition, like it determines whether the recombinant protein is appropriate for the measurement for the endogenous analyte or not. So this is a very efficient and handy tools analysis tool which process the data very quickly and gives us the error free and the very in pure accurate analysis of the sample. So this is how the Belaja software appears. So it comes in the two different component. It comes in the USB drive, and there are a license dongle. We call them the security dongle. That is a driverless security, which provides we cannot reprogram it, or it is non-editable and non-deletable. So this all these are from the my side. Now we can move ahead with our next speaker, Dr. Ashok Ganeshan, which will give you more emphasis on our another other aspects of the multiplexing portfolio. So thank you. So if we have any questions, we can take these questions once the session is over. So thank you.
Yes.
Now,
Thank you, Dr. Deepak, for the exciting talk. And for the audience, we are also posting poll questions in the chat boxes. Please requesting all of you to please participate. And at the same time, if you have any query or any question, you can paste in the chat box itself. So moving on to our next speaker. Our next speaker is Dr. Ashok Ganeshan. Dr. Ashok finished his PhD in proteomics through UKRI fellowship, after which he served as a postdoctoral fellow in ICER Kolkata and subsequently in ICGB New Delhi. He specializes in synthetic biology and genomics. Prior to joining Merck, Dr. Ashok took on the role of product manager in Prema's Life Sciences, after which he transitioned to Avantor as a technical scientist. Currently, Dr. Ashok has taken up the role of commercial marketing manager in Merck since 2025. So, Ashok, over to you. You can please proceed with your session.
Yeah, thank you, Devalina. So, can you see my slides visible?
Yes, your slides are visible.
Thank you for confirmation. So, good morning, team. So, it's a good opportunity me to give an overview about the Multiplex research area. So, I am Dr. Ashok Ganeshan. So, I'm leading the protein pathway in Merck. So, which is having antibody, ELISA, multiplexing, and then bioactive small molecules. So, today our interest to discuss more into the multiplexing. Okay, so as a continuation with Deepak, so I'm taking as more into the application oriented. So Deepak has explained what is mean XMAP technology, what is the instrument, and what are the different instrument we have, and what is the throughput, and which is dynamic factors. So it's all discussed in earlier. So I'm continuing with the what are the panels with Marcus offering to the in different research areas. Here we have three different form we can classify in the biomarker discovery. Either the people at the beginning level, so they are interested more into the discovery, and there is a middle one is for validation, and then final for confirmation. So I can explain you what is the mean discovery. So in interest of the project, so you're taking any of the sample plasma, serum, or cell supernatant, you except the article which is stated in earlier publication, the some of the analytes present in the sample. But when coming for the cohorts from the different multicentric projects the people who are working, so they are not aware what the analyte present in the samples. So for that, the people fall into the discovery platform. So in that, what they will do, they will analyze all the analyte which is there in the sample in one sample. So Merck, what we have having, so we have a discovery panel that is called plexification screening panel. In upcoming slide, I will give you more detail about what is the included in the panel. So along with that instrument. So this is one discovery, 115 biomarkers which is available. We can go for the screening methods. Second one is the validation. Once the screening is done, your cohort study or maybe the individual project, once the analytes was screened out, then you have to go for the validation. What does it mean validation? You have to measure your analyte in your sample in picogram per ml concentrations. So here you, the kits which is supplied by the which is provided by the Merck, the Multiplex qualified assay, and we can use the either IntelliFlex, XMAP, or the LX200 instrument to measure your concentrated analytes. And finally, we'll go for number of samples increase and have logged out 5 to 7 analytes of your interest to measure in all the samples. So that is called confirmation. So that confirmation is for you can offer the low plex again. In the validation, you're going to measure all the analytes in the picogram per ml. Once it's screened out, then the target specific, you're going with the low plex. Either if you only one marker which is interested in your the study, then you can go for the confirm ELISA or Merck offering ELISA kits are available. If early stage of the disease, if you want to screen it out, then we are offering the SMC immunoassay kit, that is a femto level expressed protein in your sample can be measured. So that is we are offering in the femto level expressed protein also. So these three will be offered by the Merck. It's a proprietary kit by Merck. Okay, so coming, what is the overview we are offering? So we are offering around 225 milliplex panel as a qualified panels, more than 1100 analytes, which is we having in our analyte list. So why we need to have a Merck milliplex kits for the your biomarker discovery? So there are few advantages over here. So we are offering more than 13 different species which we are working. So mainly for the human and then primate, rat, mouse, cell signaling. Specifically, this is cell signaling. I will talk little in the later, but we are offering the cell signaling panel, almost 22 different panel we have with us. That is one unique offered by the Merck. And apart from that, what is the advantage we have with this kit? So it is customizable to shoot for your research needs. It's very, very convenient, which is offering for the premixed options are available. And additionally, if you're working for the multi-centric projects, we need a lot of kits. The high volume requirement are bulk kits. That's also we are offering. And pre-optimized kit component for minimal preparation step, because in some other in ELISA we need a lot of steps which is involved, but in our kits it is maximum 3 to 4 steps. Within that, the experiment will be completed. So now we will go in detail to discuss more about what are the panels we have with us and how it's going to address in the research areas. Okay, as I mentioned earlier, so for the screening panel, we have a flexibilization screening panel. So this one unique panel which is offered by the Merck, which is having 115 biomarker available in this panel. What are the research area which is covered in this? Mainly we have kept it inflammation and immunology specific, and then metabolism and endocrinology, cancer, and then cardiovascular disease, bone, and the neuroscience, and then toxicity. So these 7 very important disease specific which is we kept it here. For the screening panel, it is not absolute quantification, this is a relative quantification. You able to see screen your marker, like its SR node types, whether it's present or absent. So along with that, you can able to get the data for the relative quantification, how much the fold might be the expressed proteins are available. So this is how we are offering for 115 in the market. So this is one good for the people who are working for the cohorts or aging research or in the drug testing or in the drug monitoring. So all the people can utilize this panel to complete their cohort studies. So mainly this is for the very convenient. For let's take an example in the earlier period of time. Let's 115 markers are available. So 115 separate ELISA kit we have to procure to check one by one. It is taken long time process to complete one cohort studies in one sample. Think about the multiple sample and then multiple analytes. So this is one addressing in a single point. In one sample, we can able to identify 115 markers within a 25 minutes of time period. You will able to identify, which is reducing your time in your project to maximize your output. This is what multiplex plexification screening panel is offering it. Mainly people who are working in academia and pharma, biotech researcher, which is they want to relatively change between the differentiating the health and disease conditions, the target identification, the leads for the disease they can able to identify in this. As an instrument, also Deepak has mentioned it. We have four different instrument with us for the LX200, MacPic, and then IntelliFlex, and then FlexMap 3D. So LX200, which is capability for maximum 80 analyte, we can able to screen if you have an instrument or you are seeking for the support. So LX200 can extend 80 analyte in this panel. So if you want to cover 115 analyte in one go in one sample, so you have to take a IntelliFlex RUO that is have capability for 500 analyte in one sample we can able to identify. So 115 all in one sample we can do in the IntelliFlex. So this kit is we are offering in the different configurations. One is for the plexification screening pen configurable from the 2 to 115 analyte we can able to customize it. Another we are offering for the pre-mixed kits, which is for 115 for the IntelliFlex, and then 80 plex for the LX200, which is fixed. So this is mainly for the screening purpose. After the screening is done, so sorry, okay, before going that, so where are the screening is required? So which is I given the point here. So this is a flexification which is going for high throughput screening, mainly for the discovery studies, which is for ideal for the exploratory research for identifying the potential biomarker for novel targets. And then we can go for the transition to the qualified assays when before starting to the quantifying the individual analytes, just identify them in a screening whether presence or absence, then we can able to go for the targeted analyte only to the quantification methods. So this is very convenient. So industry and then labs, so the people widely they are using it in the small labs. So people already they are referring to the publication and they're sticking with the directly the analyte which they're going for the measurement. But coming for the in the different cohort related and then multicentric project and aging related things, so the people need to have screening then followed by the validations. So biotech forms and then pharmaceutical diagnostic and environmental food safety it is helpful. And when coming for the lab, mainly for the academic people who are seeking this for the high throughput screening and discovery research and translation research, preclinical research lab also, and university and colleges. So it is applicable to all the industry forms as well as the academy and hospital. So people who are seeking into the big projects, so they can utilize the expectation panel to their experiments. Okay, coming to the qualified panels. So as I mentioned earlier, we have around 225 different panel as a qualified panels, which is we can able to measure individual analytes in picogram per ml concentration in your samples. This is one panel which is well versed and well famous kit in wider. So we have a cytokine, chemokine, and growth factor panel A, and additional to that panel B also there. around total panel A carrying 48 analytes, and then panel B is carrying 48 analytes, total 96 analyte, which is specific for the cytokine, chemokine, and growth factor panel markers. So here, what is advantages we have? Suppose you are not interested in all of them in one go. Maybe if you are interested, I'm interested in either only 5 analytes or 10 analytes or 15 analytes. Here are the options, so you can pick and choose. It's a customization. So either of the analytes from this panel you are making in one kit, so that there is no performance change with the configuration, that is assured one. So this is one advantage we have. So coming majorly what we are apart from the cytokine, chemokine, growth factor, so we have a different specific panel we have, that is for cancer mainly, and then cardiovascular, neurospecific, and then aging, and then metabolism. So many different panel we have specifically for the species specifics also. We have, as I mentioned in the table, that is having all the species. So here, what is we have cancer research along with the uh, cytokine, chemokine, and growth hormone. We are offering, in addition to that, 220 biomarker, which is specific for the cancer-related research. The people who are working in the cancer, either in the pathway related or mechanism or stopping the cancer innovations. So we have a popular analytes with us with a different panels, which is classified into that immune checkpoint proteins. The general set of the people who are seeking for the checkpoint protein level, the regulations. So this we have a panel, and circulating tumor markers to identify the cancer. So generally they will taking the blood and to screen it out, what are the markers circulating in the bloodstream. So these are the common marker, and we have a panel, so we can get it out and screen it out of the cancer related markers. And then angiogenesis, and then metastasis. So we have majorly covering all the cancer related biomarker with us, which is we have classified, and we have a different panel sets. So these are the panel detail, which is in the right side. In the left side, this is the panel information. So we have classified checkpoint protein marker. We have total 3 panel with us. So this panel, which is for even oncology checkpoint protein 1 and 2, and then mouse specific also we have, and human as well as the mouse. The both we have it with us. So and then going for the circulatory tumor marker, again it is going for the human and mouse, which is classified. So end of the session we will share in the chat box, so all the analyte and the brochures in the chat to verify more and get your interested analytes. Again followed by the angiogenesis and then metastasis. So these are the panel list with us. And okay, so these are the panels. So what advantages we have? Why do we choose the Merck Multiplex panel for your research? So few component which is very important before you are choosing your panels. One, the cocktail we are offering into the kit. So this is a minimized, as I mentioned earlier, this is minimized step which is involved for the performing the experiment. So you can see the star indicated. So the thing is there. So here the controls QC1 and QC2, which is provided to comparison and then data comparison as well as additional validation can able to perform every kits when you're buying every kits which is supplied the QC1 and QC2 individually to validate your experiment. And here the bead diluent and then serum matrix, which is very unique offered by the Merck, which is none of them offered outside of the Merck. Other competitor were never offering this serum matrix and then bead diluent. Bead diluent, what it can do, it is reducing the false signals in the kids. So which is mean the antibody, because I said it's a multiplexing. We have more than 10 or 20 or 500. The antibody to antibody should not having any interaction. So that the bead diluent will be taken place to controlling the interactions and making a false signals. So this bead diluent will help us to reduce the false signals generations. And coming to serum matrix, this is very unique. It is very important when working people for serum and plasma. They or the kit is providing the serum matrix. What it can do, it is mimicking the environmental or the serum and plasma in a control and standards. Generally, we have a sample for serum and plasma, but in this control and standard, we are not applying any matrices, but we are providing a serum matrix which applied to the control and standard. It is mimicking 1 to 96 well all commonly as a serum, which is good for us to background calculations, which is very, very important. Another things, detection antibody cocktail, which is one kit to another kit, the data, a lot to lot reproducibility in any flex or any batch, you will get the same data. So that is what here we are offering. This is one of the unique, because after the screening, after the qualification, for this component, what is we can having advantages in generally in ELISA. What we are doing, either we are going for the duplicate or biological replicate. We are doing in the ELISA kits, which is sample numbers within 20 we are going it. In the biological replicate we are doing it, but here we can use single hit, because which is QC we are providing, serum matrices we are providing, and B diluent is providing, which is data point is very, very unique. So there is not recommended, or it's not mandatory to use the duplicate or biological replicate. If it is your publication related, if you want to use it as a duplicate or triplicate, you're good to go. But generally this kit is in one go 76 sample, we can utilize it in the one plate. Again, I'm saying 225 different panels, which is majorly for the human, and then rest of the animals also we have it. Okay, coming to the specialized Multiplex, which is offered, because generally we are year on year we are improving the addressing the challenges which is facing in the research or in the pharma or CROs. So we are helping them to sort it out their issues. One of the interesting panel, which is humanized mouse panel 1 assay, which is having around 41 analytes in total, which is species reactivity for human and mouse. So this is one is encrafted the mouse and the human genes, and then cells, and then tissue, which is encrafted with the mouse. So when you want to do, it is better mimicking the human immune response and disease mechanisms. So this one panel, which is having similar analytes for the mouse as well as the human, which is we can use for serum, plasma, or cell supernatant, all the three different matrices we can use it. Minimum sample volume which is required for 20 ml for the duplicate wells. For the neat sample, we required only 10 microtroph samples, good enough to test your all the cell cytokines profiling, which is coming in the 96 cell format in the overnight incubation. We can see the right side. This is in distinguishing very, very clearly the standard curves in the serum matrix. The overnight primary incubation, which is displaying there is minimal cross reactivity, except that is IL-5 and then IL-12P40, the beads, which is within control 11% of the cross reactivity, but rest all 99% of the analytes. In the two different species, mouse and then human, there is no cross reactivity within that. So which is very, very validated. The people who can working for the CROs and then clinical trials, drug testing. So the people who rely on this kit to test it out. So this is mainly for the dual species detectability kit, which is one offered by the Merck. So if you have any questions, please free to put it in the chat, or if the questions will come to fold, so you can answer, so that we can understand better what level you are carrying the concepts. Okay, so another one, which is rat and mouse TS-Catch, which is one single analyte, which is specifically for the CRO related, the people who are requested to make a TSH magnetic bead panel. What it is generally can test it out, so we can measure or identify the high TS-Catch or the low TS-Catch, pituitary, and thyroid cancer, pregnancy related, and then chronic kidney disease, which very important. So this is also we are offering by. And coming very interesting panel, which is cell health magnetic beads. So most of the people working for the cell culture related, the people want to assess the cell viability, metabolism, proliferation, and then autophagy, oxidative stress, DNA damage, and protein synthesis. So this is one very good panel to assess your cell viability when you are going for any drug testing. How the drugs affecting the cells, especially for the oxidative stress, general stress, and DNA damage, and cell cycle, and then inflammatory response. So this all 16 marker which is able to distinguish and say these are the marker which is elevating from your drug testings. So where it can applicable mainly for drug development and toxicity, which is it is able to identify the harmful component in the development process to identify how it is damaging the cells. And then cell cycle studies, monitoring cell health through the process cell cycling we can do. And coming for the cancer research and then aging research, specifically what are the cell which is not able to multiply or maybe the not able to divide, that may be determine the aging. So we can able to calculate into the aging research is very helpful with this help the panel. And finally, coming to the cell signaling panel, which is AKT mTOR. This is one unique panel which is offered by the Merck, which is mainly focusing on the cell culture related. So AKT mTOR, we are having both the panel. One is for the phosphoprotein versus total. You can able to measure in two different status. One is for the total protein versus the phosphoprotein. So this will able to correlate your cell signaling in the cancer research and then metabolomic and then aging and drug discovery, which is carrying 11 analyte, which is key pathway, key marker in the pathways. So this is one case study which is has been tested out on using the panel AKT-mTOR, which is signal, which is having provided the positive and the negative control for the cell lines, which is tested on membranes proteins, which is as proven the phospho versus total, and the intra and the intra-CV percentage within the control ranges. So in addition to that, we are offering BCL2 family apoptosis panel, and then DNA damage is very important in the pharma people who are working for the chemotherapy testing, how the DNA is damaged in the chemotherapy, and the total 7 marker which will indicate into the what are the DNA damages are occurring. So what is application generally? The cancer therapy development, neurogenetic disease, immunological studies, and toxicological screening. So this is a final one, which is in addition to that, we are offering NF kappa 6-plex protein translation, very important in the cell biological research, and then human RAS-RAF oncoprotein, and then SRC family, TGF beta DNA damage, which all are mainly for toxicology, neuroscience, immunology, and drug discovery and cancer research. So overall, coming how we are strong into the publication related. So year on year we increased, and 2025 we are standing almost 20,500 in publications so far with the reputed journals, with the Cell, Science, Blood, Nature, which all are we are rolling into that. So this is providing high standard kits. So coming to the final closing remark, what we are offering into the by Merck, we are offering the kits to you as ELISA kits and then multiplexing kits in the 225 kits. And then we have offering services for the samples. If you have a samples, if you want to test it out the biomarker, you can buy the kit and you can utilize the Bangalore office. We have lab, we have an instrument. Deepak and their teams are running regularly for the service, so you can utilize the facility to complete your biomarker search. If you want any assist or clarity, so reach out to us. We can have more discussion on the study design specifically. Either it's going for the screening or in the validation, so that you can able to screen it out. So that's all from my side. If you have any questions in the chat, please reach out.
Thank you, Dr. Ashok. That was a very insightful session. We have already posted a number of poll questions in the chat box as well. So
Okay.
I request the audience to please, interact with us. And at the same time, we have also pasted the feedback form. So we have pasted the link. So I would request all of you to kindly fill up the feedback form. If you have any queries, like Dr. Ashok, Dr. Deepak, they have mentioned regarding any of the kits or in your research areas, we would request you to kindly get in touch with us. And at the same time, if you have any questions right now, you can please raise your hand or you can type out your questions in the chat box. We'll be free to answer them. Okay, I don't see any hands that are raised. So I believe we had a very exciting session. So with this, we end our webinar for today as well as for this year. We will be posting out soon about the next set of webinars that we are going to start from the new year. And I'm hoping that we will meet you all again there. Thank you so much. Thank you to the speakers. Thank you all the attendees. Thank you.
Thank you so much for the participation. Thank you, Deepak. Thank you, Meghna, for the organizing.